New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
Hello, everyone, and welcome to the Real Science Lecture Series. My name is Kim Jones, a member of the marketing team at Balchem, and today we welcome Dr. Todd Callaway from the University of Georgia to discuss the effects of probiotics, their mode of action, and overall effectiveness. Todd Callaway is an associate professor in the ruminant nutrition and gastrointestinal microbiology in the Department of Animal Science at the University of Georgia. Todd received his bachelor's and master's degrees in animal and dairy science from the University of Georgia and PhD in microbiology from Cornell University. His research focused on the effects of ionophores on the microbial population of cattle. Dr. Callaway joined the Agricultural Research Service of the USDA in the Food and Feed Safety Research Unit in College Station, Texas, before returning to UGA. His research focuses on the role of the gut microbiota in the nutrition of cattle and how the complex environment of the cattle gastrointestinal tract can impact food safety and production efficiency. Dr. Callaway received the 2007 ASAS Early Career Research Award, the USDA ARS Early Career Research Award, and the 2023 American Feed Industry Association ASAS Ruminant Nutrition Research Award. Dr. Callaway is editor-in-chief of the journal Foodborne Pathogens and Diseases and serves as an expert panelist for the Food and Agricultural Organization of the United Nations, World Health Organization on antimicrobial resistance and foodborne pathogens in swine and cattle. Dr. Callaway, welcome to the Real Science Lecture Series. The floor is now yours.
Okay, thank you very much. And, basically that introduction is all a very long way of saying I'm just one of the nerds that works in this field and trying to understand how probiotics work with the animal and with this microbial population. And my fascination with understanding the microbial population began as a grad student when I was doing a traditional rumen nutrition study, but seeing how important those microbes were, and then trying to understand how can we change that microbial population to produce a better product, whether we're talking about higher quality beef, more milk, higher fat yield, whatever our goals were. How could we use this microbial population to actually improve animal productivity, animal health, whatever metric you want to use? How can we do it better? And at the time, my family still was raising beef cattle, and we had people coming around trying to sell us these probiotic products, and they would tell us it's going to make your animals grow better, have better hair coat, digest their feed better, their teeth will be whiter, their hair will be darker, they'll start driving your tractor for you, they'll fix flats, they'll date your daughter, all the things they would tell you. And we picked up the phrase that probiotics are magic Foo-Foo dust. And that kind of stuck for a long time because at the time, 30 plus years ago now, probiotics, we didn't understand what they did because we didn't really understand what that microbial population was and what was going on, how this interaction between that animal and the microbe worked. So that's one of the things that the past 30 years of research, and I've done a very small portion of that, has shown where we can understand that. And we, in the past 5 years especially, we've started to reevaluate how probiotics work. Now, there are some products that are still on the market from 30 years ago that are fantastic products. The ones that I always thought were magic foo-foo dust are long gone out of the industry. So there's been a lot of good science, and a lot of our old products withstood the test of time. But we've got to understand what these products are, how they work, and more importantly, why they don't work sometimes. So, you guys have all heard a lot about the microbiome in recent years and how it can do everything. And when you listen to the advertisers, especially watching football games, you'll hear them talk about how this is a good microbiome or a bad microbiome. I've done this as long as anybody. We actually started doing microbiome studies before I'd ever heard that word. We had no idea that word existed for about 2 years after our first publication on microbiomes. So, we've done this a long time, and I still can't tell you what's a good microbiome and what's a bad one, because that's a judgment that's going to depend on so many things. But we know it's a lot more important than we ever realized. When I was coming along, we knew the gut microbial population played a big role in animal efficiency, health, but we know more about food safety, carcass quality. And a lot of that's driven by research from people like Mark Light at Iowa State, where he's started to push this theory that the microbial population should be viewed as a separate organ system, like the circulatory system, the lungs, any of these other systems, because of the impact they have, especially because it's wrapped up together with endocrinology. And this microbial population is related to our health outcomes, but also very impactful on our energy utilization and nitrogen retention. Because when you start seeing too much nitrogen being washed out of the animal as urinary urea going down into the waste stream, that can start to impact the environment, and that can be very impactful, to say the least, on our producers. But we also need to know things like, how are we getting our fermentation of fiber going in the hindgut? And so understanding this microbiome, knowing what's a healthy population or what's a bad population or a dangerous one, is really important. And how do we shift more to this side of that equation? Okay, so I'm going to let you guys sort in. This explains everything in one slide. And in reality, I'm just teasing with that because I can barely read a lot of this. And this is looking actually at pigs, but this gives you an idea of what microbiome data looks like, where you see changes in certain microbial members at different phases of life, or if you do things like fecal microbial transplant, that you can shift the population. But it is really difficult as a practical nutritionist to look at data that looks anywhere like this and make conclusions other than your broad conclusions about what is the microbial population doing, what are its end products. Because these numbers, whenever you see these called principal component analysis plots, you notice how there is no label on the X or the Y axis. It's these random numbers. It just shows relative abundances or relative levels, but that doesn't tell us anything biologically. So we have to be careful about looking at these, because in this case you have a cluster here and you've got an arc cluster here. That tells us some things in the understanding of what that microbial population looks like, but it doesn't necessarily make predictions for what we're going to see in the animal. So I want to make that very careful, because this is really intricate data that is sometimes misapplied by a lot of people. So backing out to that 60,000 foot view, we've known for years in ruminant nutrition that microbial population was involved a lot in short chain fatty acids. There's been a lot of work on that, a lot of work on insulin resistance. There's a lot of work that Andrew Foote's doing out at Oklahoma State looking at the microbial population and insulin resistance, but it's all about capture of energy and nitrogen. And that's largely what we focused on for a really long time on the ruminant nutrition side. But we've started to find things with more diseases like type 1, type 2 diabetes, again, linked with insulin resistance, irritable bowel syndrome, atopic eczema in people, heart disease, leaky gut. Lance Baumgart has done a lot of great work looking at leaky gut. And that's some of our collaborations have been looking at trying to understand what causes those leaky gut cattle. My son actually had atopic eczema for years, and I gave him a probiotic, a cattle probiotic years ago. I'm not going to name names, but they know who they are. And it cured his eczema within a couple of days. Literally, it was gone, and he never missed a day of school after that. So you can have real impact on health of humans as well as animals. In humans, we've seen a lot more evidence of roles in brain function, mental health, roles of depression, manic depression, bipolar symptoms, schizophrenia, all have microbial roots, at least in part. But we really have seen a lot of increase in our understanding of the role of the microbiome in inflation, autoimmune conditions, and immunomodulation. And we think this plays a big role in our efficiency because if your system is inflamed, the animal has a lot of systemic inflammation. That is a lot of non growth energy expenditure that is being used simply for this inflammatory response. And we'll come back to that towards the end and see how probiotics can actually impact that inflammation. So there's a lot that we can do in adjusting and changing the rheostat on this gut microbiome. Unfortunately, it's not a switch. It's not a yes, no. It's definitely a dial that we can affect the host animal health and performance by altering that microbial population. Now, guys at Illinois, and this was Josh McCann, Brian White, and some of the other guys there, did a really interesting study a few years ago where they grouped dairy cows in their residual feed intake into efficient groups or inefficient groups, and they looked at the microbial populations. Now, when we look at ecologically speaking, ecologists view diversity as inherently a good thing because it provides this plasticity to an environment in case there is an environmental shift that allows populations to adapt. And ruminants, that's what they've done for millennia, is adapt. That's what the ruminant is really good at. You can change feedstuffs. The animal could find a different grazing area and get different feedstuffs and survive just fine. But that's always come at a cost of efficiency. So what they found was the least efficient cows had a very diverse microbial population that would change or would produce a wide variety of metabolic outputs or metabolites. And out of all those metabolites, only a few of them were relevant to the animal. So these were producing very few things that the animal could use. Whereas the more efficient cattle, it was a much less diverse microbial population that produced a more focused or narrow band of metabolites. But most of these were actually usable by the animal. So you got better energy capture in these efficient cattle versus inefficient. When we stop and think about it, that starts to make sense. The analogy that I utilize on this is an inefficient animal is me building a garage or building a car in my garage, not the other way around. Building a car, learning how to do every job myself. It's going to take me years or millennia to do that. But if I learn one job and build it on an assembly line, I can build a car very rapidly. And that's what these microbes are doing. You have specialized organisms producing end products that matter to the cattle. So that if we can make that shift, then we can help improve the efficiency of the animal. But that means we've got to link the outputs of these populations with the microbes that we care about, which ones are important. And in this study, they found that it was the conversion of lactic acid, our old friend of lactate, going to propionate by Megasphaera elsdennii and Coprococcus catus that was linked most significantly with this increased efficiency, which starts to make sense. You have less methane production, you've got a glucogenic volatile fatty acid. So it makes sense that these cattle are going to be more efficient and grow better. So all these things start to fit together. And that's led to a lot of the research that we're doing here currently at Georgia, trying to understand what's going on with our cattle. And it's a three-legged stool approach, looking at that live animal performance and our normal production measurements and metrics. What's the microbiome look like? What's the diet? And what are when we look in the old-fashioned microbial, the old microbiology that we did 30 years ago, looking at in vitros, but what are the VFA end products, other metabolites, ammonia, methane, digestibilities, all those traditional measures? How can we link all of these and finally square that circle to understand this interaction of diet, microbes, and live animal production to be able to improve efficiency, production sustainability, reduced environmental impact, with a better carcass quality that's safer? So how do we do all that? And that's what we're trying to do, is link all this together. Because quite honestly, that balance of the microbes really does matter. Because if you get slightly out of balance, it's going to be a tractor crash. And this guy would know that. But our carbohydrates are fermented to produce our short chain fatty acids, again, lactate, branched chain fatty acids, etc. But you have problems out here, wait on this side of the tractor, so to speak, of salmonella, which is widely occurring. Right now, we've got high pathogenicity avian flu out there. Other opportunistic pathogens that can all affect your balance on that side of the tractor. End products, indole, methane, dysbiosis, diarrhea, yonis, you name it, can result in bad things. It's not just this kind of crash, but the crash can affect other cattle in your herd as well, or in your pen. So that word that we use a lot of, dysbiosis, it's this imbalance or perturbation of that microbial balance. And it's just looking at a change, but we're used to looking at dysbiosis of like acidosis leading to laminitis, or some of the really cool work that Rand Broadway and Kristen Hales are doing, looking at liver abscesses, trying to help reduce the incidence of those abscesses. But how do we reduce subacute ruminal acidosis, acute acidosis? How do we improve our feed intake and feed efficiency? Those are really big deals. And those are driven by a microbial population that gets out of balance. So again, leading to leaky gut syndrome. And I'm sure Lance has talked to most of you several times on that, so I'm not going to go into too much on that. But that leaky gut is a link between that microbial population and a non-growth energy expenditure, this inflammatory response. But it's also a route of entry of pathogens to the animal, whether we're talking about liver abscesses or we're talking about salmonellosis in your animals, which will also impact morbidity and mortality of your animals. So there's a lot going on. It's really hard to unpack all of this. And trying to unpack it is a lot of what we've been doing for several years now across the industry. And some of our results are looking at our low efficiency or high efficiency cattle, found changes in that diversity, or what the Firmicutes to Bacteroidetes ratio, groups of bacteria, what their ratios were, was a little bit linked. And that's been something people have wanted to link for a long time. But changes in populations like of higher ruminococcus led to more efficiency in our beef cattle. They also had lower bifidobacteria populations, and bifido is linked in humans and pigs with better survivability of newly weaned animals or milk consuming animals as well. So it's probably related to more extensive degradation in our cattle, but also changes in your methanogen populations. And we'll see some of why that's important in a little bit. But you get less waste of that dietary energy intake as methane. But one of the things people will look at these microbiome numbers and tell you, well, this means X, this means Y at a ecological level. And the truth is, it's like looking at this class picture from 1983. So I'll go ahead and clue you in. This was when I was a kid, so this is a long time ago. But can you look at this picture and get anything out of it relative to what these kids' impact on society is going to be? It's really difficult to, because all this tells you is who was there that day. It's an attendance sheet. Now, if you looked at everyday attendance sheets, so you stack those on top of each other for the full 12 years of school, you'll start to see a pattern. Then we know grades are correlated with better performance and better impact on society. But that's not going to tell you the whole story. You also need to look at what jobs did they hold, what extracurriculars, what were they involved in, what was their socioeconomic status at home. All these other factors, other than just who was there in that picture one day, are going to determine those individual outcomes, but also their holistic impacts on society, or in our case of this metaphor, on the rumen and hindgut in this animal's performance at large. So just taking this one snapshot out of the rumen is not always going to tell, or the hindgut isn't always going to tell us what is a good microbiome. We're having to collect a lot of data and put it together in novel ways. So to give you the idea, there's me in the nerdy glasses, and some things never change over time. But our probiotics and prebiotics are a great tool of the trade to improve that efficiency. We can also look at organic acids and some natural botanicals, feed additives. I spent a lot of time working with ionophores, diet change, specific odd compounds with essential oils. But how can we make the animal more efficient? Because what we're trying to do is harness that native microbial enzyme pool. But we don't have a silver bullet. There's not one thing we can throw in there and say, that's it, problem solved, let's move on to the next thing. So this is a list of definitions that I kind of use, and I put this in a paper last year that came out, of looking at how we talk about these products, because people start using different terms and it becomes a little bit confusing. But eubiotics is kind of this umbrella term. Probiotics are living organisms that we add. Prebiotics are fermentable substrates that aren't used by the host animal. Now, it's hard to do a lot of prebiotics in the rumen because that rumen is really good at fermenting things. So you're using the prebiotics to alter that population, but there's a lot of bugs in there that can degrade a lot of your oligosaccharides. Postbiotics are typically thought of as non-living yeast products or fungal products or products of their fermentation. So this is a lot of what we used to call direct-fed microbials, and I'll still slip into that terminology. It's going to take a long time to retrain my brain. But symbiotics are using either pro and post or prebiotics together. So a lot of our postbiotic type products may actually contain some prebiotics in there as well. So that's what we mean by symbiotics. But also we have phytochemicals that can, or natural botanicals or nutraceuticals, compounds that we can feed to animals that are going to shift that microbial population as well. So when we include any kind of eubiotic approaches, we can work at the rumen level in that hindgut population, but we also have to consider what is our impact on host response. because everything we do is an effect on an ecosystem within that animal, within a larger biological system. So we've got to work on adjusting these populations, these two different ecosystems in the rumen and the hindgut, to drive this host population. So, we can change nutritional nutrient competition, pH, decrease methane production, increase propionate, get that nice acetate to propionate shift to increase efficiency in marbling. We can increase butyrate in the hindgut to get more enhanced gut health, increasing that last minute capture of feedstuffs, of energy and nitrogen from those feedstuffs that the animal can utilize, that's escaped ruminal degradation and animal digestion to the small intestine. Try to get a little more of that harvested in the hindgut. But also we see responses at that host level of looking at reduced systemic inflammatory response, reduced antibodies, and increased growth hormone production. So we see some really broad-ranging impacts. So looking at this microbial population and what we think probiotics do, and again, I have to emphasize, we think this is what's going on. We don't know. We know that all these things do occur. There may be things that we just aren't seeing yet. But you have in the lumen of the gut and in that mucus layer, you've got a microbial population that is resident there. And using a probiotic, whether you're talking a bifido or a lacto or somebody else, is going to produce these short chain fatty acids or antimicrobial proteins that are going to affect the microbial population in that lumen or in the mucus layer. And that's going to shift your microbial fermentation, change those output metabolites that the animal is going to get to utilize. And you can change that population pretty significantly, and you can competitively affect those receptor sites where the animal is experiencing the microbial world inside it, where those interactions happen, down on the epithelial face of the cell. Now, as these tight junctions, as their name implies, they're supposed to be really tight between cells. But without a lot of energy going into these intestinal villi and being absorbed by that epithelial cell, without that energy, those tight junctions become looser. The zona occludens, all those proteins become a little bit looser. And if you're not getting a lot of energy to the cells, you can start to get pathogen slippage through these tight junctions, which is really pretty bad. This is where the whole idea of butyrate enhancing gut health comes from, because when butyrate is absorbed by the cells, it's going to be utilized primarily by that intestinal epithelial cell, giving it more energy and helping tighten those tight junctions up, improving that gut integrity. But you also then have your immune system. Again, 70% of that immune system is located in the gut. So your dendritic cells are sampling the microbes and helping to stimulate your B and T cells and regulating that cytokine. So you can start to see how this host immune system response is linked to your gut integrity, leaky gut, and the microbial population in the gut. How all these things tie together. You can't just say this is doing X or Y. These things are doing a lot of things simultaneously. So you can look at production of VFAs, inhibitory compounds, etc. But the effectiveness of probiotics and any of these eubiotic type approaches is looking at a combination of any or all of these, where they're helping keep pathogens at bay. And they're also helping to reduce changes in that microbial population's impact on their animals. But as I mentioned earlier, it's important to know why some of these products work. It's also important to know why sometimes they don't. And this was a interesting study where they looked at many different kinds of probiotics and looked at the effect of methane and total gas production of these different microbes. This was doing in vitros. And they then came back and looked at some methane emission from sheep, fed the best of them for that were fed hay. And you can see over different periods of time, different effects from these different probiotics. So some work really well in vitro, some work less well in vitro, but will work okay in the animals. So there's more going on than what we necessarily know, but we see variance in effectiveness of the different probiotics. Further, and this was a neat study these guys did, where they looked at 3 different probiotics, in this case, Lactobacillus bulgaricus, lactopintosis, and Propionibacterium freudenreichii. But they were looking at the effects of treatment with these direct-fed microbials. So again, this is actually what we call probiotic now, on the bacterial populations of the rumen, archaea, which includes all the methanogens, and Methanomycillocalase, which is a specific kind of methanogen. And what the take-home message of this study, because looking at this doesn't really do a lot of education for us, but you notice that each probiotic results in a very different bacterial population, a different archaeal population, and a different specific methanogen populations. So you see a lot of variability in effectiveness, even within Propionibacterium, which tends to work pretty well and be pretty consistent. But these variances are very common. So what that says is we don't totally know what we don't know about how these probiotics affect that composition of that microbial population. So we don't know how it's affecting which microbes show up at school on any given day. Stick with that metaphor. And this is one of the coolest studies that I have encountered. And it's really changed my way of thinking how probiotics really exhibit a lot of their effects. So they used some lactating Holstein dairy cows, and they had one group on day 0, or 2 groups on day 0. They looked pretty similar in the upregulation or downregulation of host gene expression. So they were measuring a lot of different genes, and they found that about 11,000 genes were differentially expressed after 60 days between the controls and the probiotic fed cow. So they looked pretty similar at the beginning. They look pretty different at the end. So what these colors mean are blue means down-regulated, red means up-regulated. Well, they found 11,000 genes of the cows were differentially expressed between probiotic treated and controls. And you notice there's a lot more blue than red that you see in these. And these downregulation impacts were linked to 87 different bovine metabolic pathways. A lot of them involved with growth hormone signaling, which was increased, so that's your reds. It downregulated the inflammatory response and reduced systemic inflammation in these cattle. So it's really cool showing that probiotic treatment alone can affect your gene expression by your cow. And that's something I never really thought would happen. So this has kind of opened my eyes to a lot of changes. Now, another theory of action on this is looking at the enhancement of things like an indigenous Selenomonas or Megasphaera population, because those are lactolytic, or they will ferment lactate to form propionate. And this is something that people have been trying to harness for at least 35 years now. And one of the things that when I first came into grad school, I started working on was looking at malic acid. And this was some Scott Martin's work years ago with malic acid, reducing methane lactate production, shifting that acetate to propionate ratio. And it actually looked a lot like the effects of ionophores. And what we saw was basically it's forming or causing a shift in that either the um, acrylate pathway for Megasphaera or the reverse TCA cycle used by Selenomonas that causes those organisms to ferment more lactate to form propionate. And that propionate acts as a reducing equivalent sink responsible for decreasing methane production. So again, the effects look a lot like monensin. And some of our eubiotics, and especially our postbiotics, contain malate or fumarate because those are intermediates in the TCA cycle. So the theory is that these unintentional, again, symbiotic type approach, these unintentional compounds that are included in that product may actually be causing these indigenous Pseudomonas and Megasphaera to increase their rate of lactate uptake, shifting that acetate to propionate ratio, increasing pH, and helping reduce both SARA, subacute ruminal acidosis, and ruminal acidosis. Now, the effects of using these dicarboxylic acids, malate and fumarate, are really similar to monensin and also complementary. They work by different pathways, and you could actually additively affect your shift in the microbial fermentations. Now, a question that I've had a lot of is people start looking at bacillus as a probiotic product. And bacillus are really neat organisms. I mean, there's actually entire classes taught just on this slide of what bacillus do. So bacillus are really, they form these endospores, which are like an escape capsule for the DNA and some ribosomes. And those endospores are resistant to heat, light, desiccation, UV radiation, starvation. They've actually taken some of these bacillus spores they found in amber that are at least 200,000 years old, and I believe there's someone that's in the millions of years old now, that they have been able to resuscitate out of amber. So you've got your own Jurassic Park of bacillus where they can take these organisms and they can come back to life. They can germinate. And the advantage of this for bacillus is you can add them into pellets or a normal feeding process because you know the heating isn't going to kill them, at least not really kill them. It's going to make them go into a non-vegetative state, into these endospores. And in the ruminant gut, given enough time, those spores will germinate and then grow like a normal bacterium. But when times are bad, they'll form these spores again. But what causes them to decide to go between vegetative or sporulating is kind of poorly understood, especially in terms of looking at gut ecology. Now, some of the problems with this is some of these bacillus organisms are bad guys. Bacillus anthracis comes to immediate mind, Bacillus cereus. But anthracis is the anthrax organism. Same family, but a lot of these products are very safe. And they're really good probiotic candidates like amyloid liquefactions and Bacillus subtilis, which produces a bacteriocin, which is a protein that can kill specific bacteria. So it's killing off its enemies. So these bacillus have a really great advantage there. Again, since they're pressure and heat resistant, they're pretty amenable to our industrial application and feed production. Other things of looking at essential oils that can shift this microbial population and can increase our animals' feed efficiency are really neat approaches and really useful. Whether you're talking cattle or you're looking into poultry or to swine. But you can see you can shift just with like limonene, terpenes, or orange peels, you can change that microbial population in such a way that it's going to increase our growth efficiency and can impact that immune system activity. So there's a lot of promise in how we can adjust our microbial populations. Now, a study that one of my students recently completed, Andrea Osorio, found where we were looking at a shift in cattle going from a real high grain diet to a high quality forage. We found more than 100-fold drop in E. coli populations. Generic E. coli, we were using it as a surrogate for E. coli 0157 and some of these nasty or pathogenic E. coli. But it just shows how a shift in a diet alone can cause a really significant change in a specific population. So we need to start understanding what our drivers are of these type of changes, because that's going to help us focus on what our metabolites are that are important and how we get to those metabolites with our probiotic type approaches. But ultimately, we've got to understand the microbiome action to understand which probiotics work. We need to understand that old-fashioned biochemical concept of specific activities, of milligrams of protein used or of substrate used per milligram of microbial protein per time, and be able to connect those end products with the microbes and their impact on host physiology. And some of these organisms can do more than one thing at once. So you got to model both sides of an equation. So really, this is a really complex game of Pokemon, of understanding how these organisms work together in their environment. And we got to understand how each of these probiotic approaches that we're using, how they all work together, how they function to change that microbial population, how they affect host physiology and host immune status. And it may be eventually that we almost have to have very farm specific approaches, and definitely production stage dependent probiotic approaches, simply because of that increased complexity of the interactions between animals, their diet, their environment, and our production goals. So with that overall, our eubiotic approaches have a much brighter future today than what I ever would have thought 25 years ago. They drive production efficiency, carcass quality, food safety, animal health, you name it, they do everything. But there's a lot of diverse activity spectrum, and there's a lot of choices for our producers. They run into a lot of decision fatigue and contradictory information because there's a lot of confusion of things like indigenous bacteria versus endogenous versus exogenous. What are those benefits of different types of organisms? What are their modes of action? What do we care about? So we really need to understand how these microbes work, what their populations are, and what's a good or a bad microbiome. That's a question I really would like to answer in my career. But we've got to be able to find that to be able to select that best eubiotic for your producer. Ideally, we want to hit something that's going to be environmentally friendly and improve our efficiency. So it's a true win-win. But your quality and quantity, the goals of your producers matter a great deal as to which product you want to choose. But those microbial interactions really have got to matter to us, and understanding how these all work together is really exciting, and it's also really confusing. I mean, the more I do these studies, the less I think I know about it. And it really is an exciting era to do this microbial approach. But I definitely want to temper people to realize this isn't every answer, isn't the microbiome. I have students that try to pop that off in class of, why do these cattle change their performance? The microbiome change. But why? And those things matter. And they matter more today than they ever have before. So I'd like to thank you guys for listening to me, and a lot of my colleagues that have helped over the years. and would love to try to answer any questions that you guys may have. So thank you guys for your time and attention. I look forward to talking about this with you.
Thank you, Dr. Callaway. And before we get started answering any questions, we'd like to share a brief video, and then we'll be right back to answer all the questions that have been submitted during today's presentation.
$0.05 might not seem like much, but when it's 5 cents for every cow every day, then it really adds up. New AminoSure XM precision release methionine provides the optimal combination of cost, feed stability, rumen stability, and intestinal release to deliver the best cost per unit of available methionine on the market today. Learn how at balchemanh.com/findyourx.
Okay, as a reminder, you can still submit questions through the Q&A tab at the top of your screen. So Dr. Callaway, your first question comes from Kelly, and they're asking, how can you tell if a eubiotic product is working on your farm?
Ooh, that is a great question. One of the best ways is looking at whatever the metric that you're most concerned about on your farm. Does it shift? Do you see changes? Like if you're a dairy producer, do you see changes in your bulk test? Do you see changes in your amount of production? But Ken Griswold always brings up this point that a lot of times only a certain small subset of your animals are going to respond. And this is where you've got to try to be able to look and see where your individual group differences are. Like if you're looking at, say, your first calf heifers, or if that's who your concerns are on your farm, you've got to be able to try to granulate that data a little bit to be able to understand what those effects actually are. But it is hard. It is very difficult to get down that deep into that data.
And kind of getting a little deeper into that realm, Charlie asks, what's the best non-surgical method for representative sampling of the rumen microbiome?
Okay, well, number one, hi, Charlie. It's good to see you, bud. The best method of getting that, well, there's ruminocentesis, and I'm never entirely in love with that because you go in and you jam the needle behind that rib, and vets like to use that because it's easy. The one we've been using is we've got a pump system, and we'll go down the throat, and we'll take a couple of liters of rumen fluid out and just throw it away because we're trying to make sure we're clearing any of the saliva, and it allows the weight to slide back into the rumen. But it's really hard to get a true representative sample throughout the rumen in that live animal. So, I mean, Charlie's question is really good, and it is a really hard thing to get to. So, if anybody's got a better idea than that, I mean, there's people that have the sampling capsule that they put in that it takes a little bit out of its environment. But I don't know when it's in the rumen, when it's gone down into the hindgut, there's not necessarily a time factor that I can read on those. And I'm still a little spooked by those.
Okay. The next question comes from Isan. Says, thank you for your presentation. Some people use bakery strains of Saccharomyces cerevisiae in cow diets. Does it work? And does strain really matter?
Some of the bakery strains do look like they work, especially from what I've seen. And again, limited window. So, this is not the gospel of the world on this one. But sometimes they work, especially in those higher grain diets. And yes, strain matters. Oh my God. I mean, not necessarily its source, but like with Ruminococcus albus, which is just a normal fiber degrading bacteria. Albus strain 7 does a lot of fiber degradation. It degrades cellulose, but it doesn't touch hemicellulose. Albus 8 degrades hemicellulose, but not cellulose. So even though they're almost genetically identical, there's something in them that's different where they have different roles of degradation. So when you start looking at, I want to choose this strain of bacteria versus that one, strain usually does play a pretty big role. And the truth is, a lot of these choices of which microbes people are going to use in a probiotic is made by simply, is this bug on the generally recognized as safe list? Because if it is, I can just pull it off there and plug and play on that pretty quickly. Whereas if it's not one of those grass organisms, it may take me years to decades to get it approved and get it into the market. So, sometimes these bakery strains will work, and Saxarvacii being a yeast has some different properties than, say, a probiotic organism of um, if you're using a live bacterial population. So there's a lot of subtleties there, but yeah, that Saxarvacii, some of them are really good. So yeah, and not to give you the it depends answer because I hate that, but it depends on
Yeah.
what the organism is and what you're doing to them.
Well, and here's another question coming in from Ryan that says, how do you choose which product or combination of products to choose from with all the options available, such as yeast, live or yeast culture, organic acids, essential oil, toxin binders, etc.?
Yeah, and
Another, it
again,
depends.
yeah, well, another it depends, and truthfully, you're going to have to play with it on your farms
Mhm.
because there's going to be some things that work in Minnesota or North Dakota that aren't going to work in Texas or Georgia simply because of those differential environments. And some of those toxin binders that we need here in the South because we got so much more aflatoxin may not be as useful up north. So you've got to look at, what these things do. That's where I come back to the idea of understanding those modes of action. One of the things that I always kind of use as a rough idea is looking at companies' R&D versus their advertising. We all know there's some companies out there that they're 100% advertising, 0% R&D. Those products probably are not going to work as well as some of these companies that are doing 70% research, 30% marketing, 10% marketing, those kind of approaches. But you're going to have to test and see what you find works in your region and your animals best. And I think you're going to wind up finding that using several of these products will be able to help fine tune that rheostat, but also will help you adjust to those insults that happen when you have a big storm system come through, when you have your cattle get stirred up by thunderstorms or a cold snap or a heat wave, that some of these will be able to help act as those shock absorbers. But you also have to think about, if I'm going to use an ionophore, do I want to use this kind of probiotic? Because those work against each other. So you have to understand what you're playing with. And it's a really great question because there's not an easy answer. And I really wish there was. And I really wish that I wasn't saying you'll figure it out because effectively that's what I'm saying because we just don't know. And that's the challenge. I mean, we know more than we did, but we're still not to that point. There's several products that I trust completely and that I'll use in my cattle at different stages, but I wouldn't use the same ones if I was feeding cattle when I was still in Texas.
Mhm.
So it just depends on your conditions and what your experiences have been. There are some people that will use those same products that I have and they swear those don't work. And I swear they do. So it just depends on what your personal experiences and your animals are doing.
And
And
with
I
the
hate to
difficulty
lay
of
that.
testing, yeah,
Yeah,
Charlie
exactly.
pointed out.
Yeah,
So that makes it difficult without being able to see it. So hard to refute the foo-foo dust with
exactly.
that kind of, yeah. Okay. Next question. Javier is asking, do you have to feed probiotics on a daily basis to dairy cattle?
Yeah, that tends to be at least what my thought and recommendation would be, if not necessarily every day, but it's simpler to do that than doing every other day. But Paul Weimer and the guys at Wisconsin have done some really great work over the years, um, looking at where they exchange rumen contents between cows. So they took high performing dairy cow, low performing dairy cow. They took with rumen cannula. They took the shop vac and they pulled all the rumen contents out of one cow and the other cow and then exchanged them. And within 19 days, those animals, for the most part, had gone back to their normal microbiome. So without keeping that constant external pressure on that ecosystem, it's going to return to its normal. So by using these probiotics daily, I think it will help keep that pressure on, or it does help keep that pressure on, to keep that new microbiome in place, because you're not going to completely replace it with all lactobacillus. You're just going to increase your lactobacillus population, and that's going to cause other shifts. It's like throwing a rock in the water. You'll see these waves going.
Mm-hmm. Here's an interesting question. Khalid is asking, if we administer poultry-derived probiotics to ruminants, do you believe they will establish colonies and function similarly to those isolated from ruminants?
Well, the probiotics don't know where they came from. Number one, those organisms don't know. But usually the organisms derived from one species tend to work best in that species. So it's possible that a poultry derived product will work in cattle. The odds are a little bit against it, because if the organism was best fit to survive in the rumen, well, it would already be in there. And I mean, I know that's a simplistic worldview. It doesn't mean it's not worth looking at, but it's going to be harder for that organism to get a foothold in this completely different ecosystem than what it evolved to thrive in. Because that rumen, yeah, it's an anaerobic environment. The gut is the gut, but a ruminant gut versus a pig versus a chicken gut has very different environmental drivers, and evolutionarily speaking, environmental drivers over time that have selected for certain skill sets by those microbes, not to get all Liam Neeson on you, but they have a very specific set of skills to survive in that crop or in the diverticulum in the bird or in the ceca of that bird, compared to living in that rumen population or in the colon of a pig. So it could work, but I wouldn't hedge my money on it without a lot of investigation.
Sure. Next question. Louise is asking, what is your opinion on using probiotic products to treat clinical health conditions such as scours in calves, which I've seen products marketed for?
I love it. I have used a lot of different probiotics in calves. I have a lot of people call me and go, "Hey, I've got calves with scours, and I've got this product on my farm. Will it work?" Probably, possibly, but there are several of those that work really well on like stopping scouring calves. So that I feel has a really great opportunity to work with those calves, because that calf is in such a dysbiotic situation anyway. I mean, their world is really screwed up, especially if you're talking dairy calves. They're drinking milk, they're getting that fermentation started in the rumen, but it's not fully developed. And there's a lot of weird things going on. So if you can put a microbial population in there that's going to settle things down or may help stop some of these opportunistic pathogens, it's a good thing. So that's an area where I think we can do better with our calves with that. And it may be worth it to, do that prophylactically in some of our calves.
Okay, we're coming up to the top of the hour. I got a couple more questions we can try to get in here. Profula is asking, saying, thank you, doctor, for your insightful presentation. How do you compare postbiotic solutions with similar strain probiotic, like postbiotic fermentation blends made from Bacillus subtilis, and probiotics, Bacillus subtilis for cows?
That's a good And I don't know that you're comparing apples and apples with that. I mean, you're comparing apples and oranges. For that, I would look a lot of cases at what are the inflammatory system markers like C-reactive protein and some of those other markers of inflammatory response. If you're looking comparing, same strain, probiotic, postbiotic, it would be interesting to see what the difference in those inflammatory markers are, because both of them will cause a shift in your microbiome. It'll be a different shift. And looking at that inflammatory response may be able to help you settle that out, because I think that's where we would see more impact and more shift.
Okay. One last question to close us out. Kevin is asking, based on the lactation cycle of dairy cows, pre, post, high, low lactation, etc., what would be the best time to add prebiotics to get the most out of it?
To add a prebiotic in a dairy cow, the best time, I would say during that run up into transition period, because that's when when you've got these cows balanced on that knife's edge, and I mean, listen to the stories of Ken and Charlie and some of these other guys that are, actually do feed these cattle on a day-to-day basis, where they're on such an edge that anything that goes wrong in those cows in that transition period is going to have such a long-term impact, that if we can help use something to stabilize that microbiome in that, even in late dry running up to transition, can really help stabilize those animals. So if I wanted to use something like that, that would probably be the point where I would bring it in. Plus, your rumen is smaller in that period because your calf is so large in there that your rumen volume has shrunk a bit. So at that point, you have to feed less probiotic or prebiotic in there to get it in there. But your prebiotic is going to be feeding a lot of microbes anyway.
Well, perfect. We've kind of run over the top of the hour by a few minutes here, so we'll call it there. Thank you, Todd, and thank you, everyone, for attending today's webinar. If you do have additional questions, please submit them to anh.marketing@balchem.com. The Real Science Lecture Series continues with educational topics each month, including our next one on July 9th, where Dr. Tom Overton will present Turbocharge Your Fresh Cow Diets. Our full summer schedule of webinars is now available online. Visit balchem.com/realscience for details on all the future webinars and to register for upcoming events. Balchem's podcast series continues to offer a deeper dive into our webinar topics. Log on to your favorite podcast platform and search for Real Science Exchange, or visit balchem.com/podcast. On behalf of Balchem and Dr. Callaway, thank you for joining us today.